Hello everyone. Thank you for coming to our tutorial on fine-grained interpretation and causation analysis in deep NLP models. My name is Hassan and I will be presenting this tutorial along with Narine, Fahim and Nadir. Narine works at Facebook AI and the rest of us are from the Qatar Computing Research Institute. Before we begin, here is a link to all of the tutorial resources. You can find the slides, a video recording as well as the practical course session here. Let's start with asking a question. Why are we interested in interpretation? Deep models have achieved state-of-the-art performance on most of the tasks, and now they are used as a de facto standard to solving NLP problems. However, despite the benefits, their opaqueness is a major cause of concern. These models work as a black box, and it is not clear what they have learned. Interpretation of deep models is important for several reasons. First, it gives us a better understanding of these models, which is important to design improved models. Similarly, we want to deploy these models in real-world scenarios, and having better understanding of the inner working of our models increase our trust in these systems. This also leads towards having ensuring ethical decision making. For example, fairness of our model. We want to make sure that our model is not making a decision based on certain biases. Okay, so what is interpretation? So interpretation is what the knowledge a model has learned and used to solve a problem. And it tries to answer the questions like, is this knowledge similar to what humans rely on? For example, linguistic knowledge, morphology, syntax, semantics, or is it completely unexpected or intuitive? This can further lead to more fine-grained questions. For example, how is knowledge distributed across the model components? What knowledge learned within the model is used for specific predictions? Does the inhibition of specific knowledge in the model change predictions? And how do different modeling and optimization choices impact the underlying knowledge that the model learned from about the task? A number of tutorials in the last few years have touched upon interpretation from different and similar angles. So here we just list them down with an imprecise description of the topics they cover. They serve as a very good reading material on interpretation in general. And later I will clearly state in slides what we are bringing in addition to these tutorials. So consider a neural network model for sentiment classification. And here it consists of several layers or blocks in case of transformer models. The model takes a sentence as input and predicts its sentiment. So previous tutorials have looked into representation analysis, which look at the entire layer and try to answer the question, what kind of linguistic knowledge has learned in this representation? And it can highlight, for example, that morphology is learned in lower layers and syntax or semantics is learned in high layers. So in this tutorial, we will go deeper into each representation, and we'll do a fine-grained analysis of neurons in each layer. And we'll try to answer the similar question, what kind of linguistic knowledge has been learned in representation, but we will use it as what kind of linguistic knowledge has been learned in individual neurons or a group of neurons. For a precise comparison, representation analysis shows what is encoded in the representation, while neuron analysis goes more fine-grained and says how this knowledge is encoded in the representation. Okay. Then another line of work that has been widely covered in other tutorials is input feature importance. That is the role or the contribution of various input features, like here these are the words, towards a particular prediction. For example, in this case, this example shows that variable has a major contribution in predicting this sentence as a negative sentiment. So while feature importance is effectively highlighting the role of input words towards a prediction, it doesn't tell about the role of neurons in making that particular prediction. So in this tutorial, we will look at the neuron importance with respect to the prediction. In short, what is the role of a neuron in making a particular prediction? Again, to compare exactly with the feature importance shows what input features are important with respect to a particular prediction, while neuron importance shows what neurons are important with respect to a particular prediction. And we call it causation, refer to it as causation analysis in this tutorial. Okay, formally, neuron level analysis unfolds the knowledge learned in the representation. And it answers questions such as how the knowledge is encoded in the representation, how various concepts are learned among neurons, which individual neurons or group of neurons are critical for the model, which neurons play a role in making a prediction. One common question that arises about neuron analysis is what can be done with neurons and whether there is any useful information on applications that can be derived from or by analyzing individual or group of neurons. So here let's take a quick look at what researchers have found and done using neuron analysis before diving into the actual methodology. So here researchers have shown that neurons capture specific concepts and are responsible for looking at very specific words. For example, here neuron 108 is looking at words related to some legal domain. And here the other neuron 711 is looking at auxiliary verbs with a negation as part of the phrase. So this is another interesting one where researchers have found that neurons act as a switch, activating in one direction for certain concepts and in opposite direction for semantically opposite concepts. For example, here a neuron switches between past and present tense on the two end of the activation space. So here, using neuron analysis, authors show that a large number of neurons are redundant in pre-trained models. For example, using BERT as a feature extractor, they were able to remove 85% of the neurons, while still maintaining the same average accuracy on several downstream tasks. And this also motivates towards the potential of creating distilled models for downstream tasks where we may not need all of the neurons of the original model to achieve state-of-the-art or good downstream task performance. So here, authors were able to show that some neurons learn spurious correlations. For example, this neuron 39 activates whenever a word nobody appears in the hypothesis. And this can be very useful to create adversarial examples or strengthening the model further by fixing the input data, because this clearly highlights the issues with the input data. So some of the related work have also used causation analysis and computed the importance of input words by aggregating over all neuron importances for every token. And this results in a much more clear and faithful saliency analysis with respect to input features. Knowing the role and importance of neurons enable various other applications beyond interpretation, such as model distillation, domain adaptation, model manipulation, architecture search, and generalization. And we will touch upon some of these in this tutorial, especially in the findings section. Let's look at the roadmap. As I mentioned before, so there will be two parts in the tutorial. One is fine-grained analysis, another one is causation analysis. So in the fine-grained analysis, we interpret how various language or task-specific concepts are learned and represented within neurons in the network. And we refer to this class of techniques as concept analysis in this tutorial. Then the second part will focus on causation analysis, which highlights which neuron in the network were used in making a particular prediction. And we refer to this class of techniques as attribution-based methods in this tutorial. In the last part of the tutorial, we will also discuss how to connect these two methods. One is bringing neurons with respect to a concept, another is bringing the importance of a neuron for a particular prediction, and how it leads way towards concept-based interpretation of models prediction. For both of the analysis types, we will look at the methods, evaluation techniques, and practical. Additionally, for concept analysis, we will also summarize the datasets used for concept analysis and some of the findings from the literature. Okay, so let's clarify some of the terminology. What is a neuron in our case? So neural networks may have many components such as attention heads, blocks, sublayers within layers, gates, cell, etc. So throughout this tutorial, we consider a neuron to be any output dimension from any of these components. For example, in the case of BERT, an output block of BERT has 768 neurons. The feed-forward neural network within a BERT block has around 3,000 neurons, and an attention head in BERT has 64 neurons. And in the literature, different terminologies have been used for neurons, such as expert, units, and features. Okay, another terminology is about concept. I have been using this term a lot in the initial slides. So a concept is an intrinsic property that we would like to probe in a model. And it can be of various granularities. Example of concepts can be gender, ethnicity, religion, parts of speech like noun, verb, adjective, some semantic concepts, lexical concepts like words, morphemes, phrases, and even non-linguistic concepts like brackets, emoticons. So we want to kind of say, given a concept, whether our model, which neurons of the model has learned this concept. For example, adjectives and negation are important concepts to solve sentiment classification tasks. So we want to question, did the model learn these concepts? And then using the concept analysis, we can identify neurons which are learning our concept of interest, which are adjectives and negation in this case. Now that we have set the foundations and terminologies, let's get started with the first part of the tutorial, which is concept analysis. So concept analysis methods fundamentally operate on activations from a given model over a dataset. So consider here a simple model with an embedding layer and an RNN layer having 3 dimensions. So word level activations will contain 6 numbers for each word in this case, 3 from the first layer and 3 from the second layer. And here yellow represents the first layer and red represents the second layer. And each of these individual units are the neurons in our case. So in addition to word level activations, if we want to have sentence level activation, it will also contain 6 numbers for each sentence. Again, 3 numbers from the first layer and 3 numbers from the second layer. So there are several ways we can extract the sentence level activations. For example, in the case of transformer models, we can use the CLS token representation. In the case of RNN, we can use last token in left to right RNNs. One thing to be cautious about when using the pre-trained base models for the CLS representation as a sentence representation, because base model CLS representations are not optimized for sentence representation. So if we want to use the CLS representation, we have to fine-tune the model on a sentence level task, and then we can use the representation of CLS for sentence level activation. But there are other alternates as well. For example, we can aggregate over word activations using average or max pooling for each neuron in order to obtain sentence level activation. Okay, so here is just a more detailed roadmap of concept analysis. We'll look at various methods and various evaluation techniques. Then we will summarize the findings, data sets, and we will show some practical. Okay, let's look at the first method, which is visualization. So from the simplest way researchers have analyzed neurons is to visualize its activation over a set of sentences. And patterns can be identified over a set of sentences to discover the role of neurons. For example, here you can see a neuron is activating mainly on a comma, and by visualizing it, you can identify the role of this particular neuron, that this is a comma neuron. Similarly, this is an article neuron. And again, with the visualization, you can also look at neurons which spread across different words or entire sentences. For example, here, this neuron is a position neuron which activates have a high activation on one end at the start of a sentence. Then in the middle, it becomes 0, and then in the end, it again goes in the other end of the activation. Okay, so since in our current models we have a lot of neurons, so visualizing all these neurons is cumbersome. And researchers have found various tricks how to reduce the search space by only visualizing saturated neurons, or by looking at neurons which has high variance or low variance. Or in the case of ReLU, you can ignore dead neurons. And in this way, you can actually reduce the search space of visualize how many neurons you want to visualize manually. But still, this comes as a limitation, because Even if you reduce the number of neurons, still there are a lot more neurons that you have to visualize, especially in deeper and deeper, when we go in deeper and deeper models. So another limitation of visualization method is its difficulty to analyze polysemous neurons. And polysemous neurons are the neurons that are capturing more than one property. For example, here, is this a verb neuron or a year neuron? Because we can see that it's activating on verbs a lot, but at the same time, it's also activating on year and the different end of the activation. And these types of neurons are very hard to visualize manually. Another issue with visualization is that it's very subjective, and researchers can be prone to confirmation bias. For example, in this visualization, one may assume that it's a verb neuron because you can see that it has strong activations on several verbs. But if you also see more closely, you may also be able to see that it's also happening a lot on different nouns and also on some year. So, but depending on what a person is looking at, maybe he has a prior that I want to look at verbs, and that's why I'm artificially creating my examples that have those verbs, and then he will right away see that, oh, these neurons are occurring on those verbs, and that's why the role of this neuron is to capture this verb, and this can easily lead towards misleading results. Lastly, there are many neurons which are looking at uninterpretable concepts, and with visualization we can't do much about it. So far, we have seen visualization as a way to manually identify the concept a neuron is learning. The limitations, as mentioned before, is that the search space is quite large, both in terms of the neurons and the number of sentences in the corpus. So the second class of methods that we are going to look at, they work on some intelligent selection over the corpus and make the overall process of neurons analysis much more efficient. So corpus selection methods either identify sentences that maximize a given neuron's activation or identify salient neurons based on some statistics computed over all sentences in the input corpus. We divide them into three categories: corpus rank, corpus generation, and mask-based neuron selection. In corpus rank, for a given neuron, we rank all examples in the corpus by their activation value. Now we have a sorted list of sentences with highest activation on the top, so we can manually visualize, we can select the top sentences and manually visualize them to identify a concept encoded by that neuron. For instance, we can find words that maximally activate a neuron and analyze its top K context from the corpus. In this example, you can see a certain pattern of activation when observing a particular concept, like the phrase next to determiner in this first example, and the words representing number 2, 3, 2 in the second example. So in corpus rank, what we were doing is we were manually identifying concepts by looking at a set of sentences. But manual identification is still cumbersome. So one way to resolve is to split the corpus into lexical concepts of various granularities, such as words, phrases, or sentences. And then we can create synthetic sentences that represent each of these concepts. And the neurons that activate those synthetic sentences will be the ones which belong to that concept. Consider an example here. So John is sitting in the car. We can break down into several concepts. For example, based on a parse tree. So in this paper, they consider each node of the parse tree as a concept. So for this example, they can have a concept pool, which is John is sitting in the car sitting in the car. So it contains all the words and also phrases till the sentence level. They also consider morphemes in this paper. So for each concept, we can create a synthetic sentence of every sentence length representing the particular concept only. Here, we want to find the neuron that belong to the concept the car. So we create a syntactic sentence, the car, and we identify a neuron that maximally activate on the syntactic sentence. And that neuron belongs to that concept. Similarly, if we have even a larger concept sitting in the car, we can do the same, and we can identify neurons that belong to that particular concept. Okay, so corpus rank has a few limitations. One is that our analysis is limited to the space of a given corpus. What if a neuron represents a very diverse concept which is not present in the corpus that we are using for the analysis? Similarly, like neuron behavior on unseen or ungrammatical sentences will be unexplored in this case. So gradient accent is one way to resolve this limitation. So instead of selecting sentences from a corpus, gradient accent generates synthetics and inputs that maximize activation of a given neuron. And it has been very successfully applied in the vision domain to generate input images that maximize the activation of a neuron. And this is an image taken from the vision where we can see that the particular neurons are responsible for particular digits from the synthetic inputs that were generated automatically. So these are the automatically generated synthetic inputs. One limitation of gradient accent is that it cannot be directly applied to NLP because of the non-continuous inputs. But gradient accent with Gumbel softmax solves this issue. So it outputs a spiky 5-heart vector representation that maximally activates a given neuron. And here you can see, first one is the corpus rank, and it shows the highest activation given a 5 gram. And based on that, one can conclude that this might be a horse neuron. But with the Gumbel softmax, they were able to generate those examples. And there you can also see that motorcycles, enthusiasts, horseback, horses coming. And this gives a more clear relation of the neuron with a race neuron. It's not just limited to a horse, but it's a race neuron. So Gumbel softmax is quite helpful in generating these kind of unseen patterns that a neuron is looking at. Yeah, one limitation of Gumbel softmax is that there is very limited work done on it. We haven't found very much exploration in the NLP domain. And it is very likely that the generated words or phrases may not make sense in the real world. Okay. So the techniques that we have seen so far have often involved a human that has to analyze the outputs of the methods to figure out what concepts are being represented by the neuron. Now we will look at some other methods that take concept data sets and try to automatically make a connection between the neuron and the corpus and the concept. So one such technique is mask-based neuron selection, where we create masks of all of the neurons and concepts and compare these masks to define the function of a neuron. And then you can use different evaluation methods like interaction over union to compute the overlap between a given neuron and a given concept, and then rank the neurons for a given concept based on the value of IOU. To be more concrete, we create a neuron mask, which is a binary mask of each neuron based on some threshold on the activation values across the corpus. Then we create a concept mask, where a binary mask of each concept based on its presence or absence across the corpus is created. Then we have these two masks. We can do an intersection over union to find which neuron mask relate the most with the concept mask, and then we can rank based on that value. So let's look at a very specific example where we have 4 sentences. We first create masks for all of our concepts that we want to consider. So here, for example, one of our concept is noun. So we will see if noun appears in those sentences or not. And based on their appearance, we will say 1 or 0 as a binary mask. For example, here, train and Paris are noun. It will become 1. There is no noun. It becomes 0. And similarly, we have 1 for cars and for the last 2 examples. Then we have another concept, which is verb. Again, based on whether verb exists in the sentence, we can put a binary mask. Then Paris is also a concept. So here we are also trying to show that you can have part of speech tagging as a concept, but you can also have word level lexical concepts as well. So here Paris is a concept. Now for the neuron mask, where we can have a mask based on the threshold on the activation value. So if the activation value is greater than 0, we consider it 1, otherwise we consider it 0. So now we have neuron mask and we have concept mask. We can create an intersection over union, and we can see which of the neuron refers to which of the concept. For example, here we can say that this neuron 1 is more of a noun neuron, and neuron 2, second neuron, is more of a verb neuron. So there are other works which have still used the mask-based neuron selection, but instead of using binary mask for the neurons, they use activation values as a prediction score. So they compute average precision score per neuron and per concept, and rank neuron by their average precision score with respect to the concept. So overall methodology will remain the same, but only the final calculation will change to average precision. That's all from my side for now. Now I will hand it over to Fahim, who will cover the rest of the methods.
Thanks, Hassan, for the introduction and for getting us started with the methods. The methods that we have seen so far have largely looked at neurons from a data perspective, relying on sentences from a corpora or automatically generating them. Let us now look at a class of methods that are more modeling based, specifically neuron probing and unsupervised methods. Let's start with neuron probing. So neuron probing methods aim to use some kind of classifier to probe representations and neurons for specific concepts using supervised learning. Specifically, given supervised data for some concept, let's say the concept noun, we first extract the activations of all neurons with respect to input words. Next, we train a classifier using these extracted activations as input features. And lastly, we look at the internals of the trained classifier to derive a ranking across all neurons based on their importance with respect to some concept. So various authors have used different classifier models and settings to derive these neuron rankings. For example, Dalvi et al. Used a linear classifier and looked at the classifier weights of each input feature to rank the neurons. Recall that each input feature corresponds to a neuron in the original model that is being analyzed. And so the higher the weight for a given input feature, the higher its corresponding neuron's position will be in the final ranking. So looking at this more formally, the feature weights here are being used as a proxy for the importance of a neuron. So in this case, the choice of regularization used for the linear classifier while training the model directly affects the weights and therefore directly affects the final neuron ranking. Several regularization settings have been explored by various authors, and we will take a look into some of them now. So Radford et al. Used L1 or lasso regularization. What this forces the classifier to learn is very spiky weights, indicating the selection of very few specialized neurons with the importance of majority of the neurons set to 0. This can be helpful if the goal is to identify very focused or very specialized neurons that are responsible for very specific concepts. This can also be seen in the figure on these slides. The lines represent the classifier weights of all neurons, with the highest neuron weight in the beginning and the lowest neuron weight towards the end. The green line, which is the lasso regularization in this case, quickly goes to 0, with very few neurons having some non-zero weight. On the other hand, Lacretz et al. Used ridge regularization, which is L2 regularization, to encourage grouping of input features. This translates to discovering groups of neurons that are together responsible for a single concept, instead of looking for singularly focused neurons. And then finally, Dalvi et al. Use elastic net regularization, which combines the sparsity effect of L1 and the grouping effect of L2. This helps the classifier takes groups of highly correlated features into account while also considering important focused neurons. Another detail that is considered by some authors is to use absolute weight values from the classifier instead of raw weights. Ignoring the sign of each weight value enables looking at neurons with both positive and negative weights equally, which can be useful for the analysis of some models. In contrast to using a linear classifiers, some authors like Valipur et al. Have also tried to use a random forest classifier to derive neuron ranking with respect to a given concept. So random forest classifiers inherently have a way to extract the importance of each input feature by computing the purity at various nodes in the trees. And hence a ranking very similar to the linear classifier can be created using this importance score. So having seen that these classifiers can indeed be used to extract neuron rankings, it is also essential to consider some important factors that directly affect the output of these methods. After training the classifier and before extracting the ranking, we first have to ensure that the probe itself is trained properly and is indeed learning the correct relationship between the neurons and the concepts. A lot of the techniques used to ensure proper training of the probe are not all specific to neuron selection, but are rather general machine learning concepts. So, for example, one very standard metric for ML models is to compare the majority class performance with the classifier's performance, and see that we are doing better than the majority class performance. So, in the next few slides, we will take a look at a few more of these techniques that can help us become more confident in our probe's output. So, let us start by looking at randomly initialized representations. Depending on the concepts being analyzed, a classifier may be able to sufficiently extract enough signal from a large number of neurons and achieve high performance, regardless of whether the concept is actually represented by any neuron. This can especially be the case if the training data used to train the probe is small, or if the concept being analyzed is very simple. Training a probe with random representation as its input can help us detect this issue. If the random probe's performance is as high as the original probe, we cannot trust the original probe to extract a ranking. Another method to test our original probe is to use a control task. Hewitt and Liang have introduced this concept of a control task, which aims to break the linguistic relationships between the words in a task. Essentially, each input word is associated with a random class instead of its true class. And a probe is then trained on this controlled data set, and its performance is used for evaluation. Specifically, Hewitt and Liang define selectivity, which is the difference between the original probe's performance and the control task probe's performance. The higher the value of the selectivity, the better our original probe. To summarize, one has to make sure that the original probe's performance is higher than all of these auxiliary tests, majority class, random representations, or even control tasks, to ensure that trustworthy ranking of neurons is actually being obtained. Apart from these techniques to evaluate our probe's trustworthiness, there are also some limitations that one needs to keep in mind when using neuron probing. Since this class of methods uses supervision, we need to make sure that we have well-defined concepts, and we also need to make sure that these concepts have good annotations. The specific probe and settings used also directly affects the ranking, as we saw with regularization. And although some authors have tried some variations, more exploration is still necessary to find the perfect probe given a specific concept and a specific model. So keeping in mind some of the downsides of the previous methods, let us now look at another class of techniques that have a weaker reliance on annotated data, namely unsupervised methods. Recall that the previous methods, especially neuron probing, relied on well-annotated corpora of concepts to extract relevant neurons. These corpora usually require very careful and expensive annotations, which may not always be feasible. Unsupervised methods aim to work around this need by surfacing underlying knowledge patterns without any supervised data. These underlying patterns may not always correspond to human understandable concepts, but researchers have found some success in discovering meaningful neurons. One important thing to remember is that because the concepts are extracted without any ground truth labels, one usually needs to further analyze them either by having a human in the loop or by using some weak supervision to automatically assign each of those patterns some human understandable meaning. This class of methods also aims to answer slightly different questions in some cases, such as what neurons are most important for a given model, regardless of any specific concept. So a very diverse set of methods have been used to identify important neurons in an unsupervised fashion, and we'll take a look at some of these now, starting with ablation methods. Ablation methods are arguably the simplest of the unsupervised methods. The key idea here is to mask or erase some neuron or groups of neurons and compare the drop of performance when compared to using all neurons without any masking. A common implementation of this erasing of these neurons in a network is to clamp the neuron's value to 0 or its mean value on the given corpus. Both of these methods effectively reduce or eliminate the neuron's function in the network. So one way that has been proposed for performing ablation is to consider only a single neuron at a time. For example, Lacretz et al. Used this technique to analyze neurons in a model where they ablated exactly one neuron at any given time. They then ranked all the neurons based on the decrease in confidence scores of the models when ablating each neuron one by one. Lee et al. Presented a slightly modified version of single neuron ablation, where instead of comparing raw confidence scores, they compared normalized confidence scores. This may be a better metric because it's independent of the original model's performance and may give a better sense of the relative importances of neurons. Now, although single neuron ablation is a straightforward technique to find important neurons, it may not always be an effective method. The effect of a single neuron may be negligible in large models because information is generally redundantly learned in these networks. There can also be groups of neurons learning a concept together, and these kind of groups cannot be identified using single neuron ablation because each neuron is looked at completely independently. So to overcome these limitations, some researchers have explored multi-neuron ablation using greedy search. The proposed method ablates neurons one by one, adding the neuron with the largest performance drop to the set of neurons to be ablated. Now, although this method does work slightly better, it is not clear if the greedy approach to adding the neurons is effective for all models. The order of ablation is critical in order to get a good overall ranking, and trying more permutations is necessary. However, trying all permutations itself is also an intractable problem, so some sort of balance has to be found here. Also, since neurons are added to the ablated set greedily, the interplay between neurons that work together in a group is also completely ignored in this case. So that was ablation. Let us now look at another type of method, Gaussian-based probing. The fundamental assumption in this method is that neuron activations follow some prior distribution, and this can help us better analyze the function of these neurons. Specifically, Hennigan et al. Assumed a Gaussian prior on these neuron activations. So they were able to find several neurons whose activation patterns match this assumption quite well. For example, in the figure we can see that the orange activations representing a present tense neuron and the green activations representing a past tense neuron both follow independent Gaussian-like distributions. Armed with this assumption, the proposed method first fits a multivariate Gaussian using all neurons in a dataset. Next, individual probes for any single neuron or groups of neurons is extracted from the multivariate Gaussian, and these probes are then analyzed to extract interesting concepts. One thing to keep in mind is that the assumption of a Gaussian prior does not always hold for all neurons, and hence some neurons cannot be analyzed using this method. Secondly, groups of neurons still need to be analyzed in a greedy fashion, which makes the search for interpretable neurons and concepts quite expensive. So next, let us take a look at some methods that directly target groups of neurons. We will first take a look at matrix-based factorization methods. For those that are new to matrix factorization, it is a class of algorithms that tries to find optimal strategies to break up large matrices. Recall that neuron activations over a dataset can also be represented as a large matrix, and breaking this large matrix optimally results in a smaller matrix of factors, where each factor represents a group of neurons that performs similar function. Matrix factorization based methods have largely been used in computer vision models, but are now slowly making their way into NLP as well. To understand this method better, let's consider this visualization. Consider the matrix on the left, where all neurons from all layers are stacked as rows, and each column represents a time step. Passing this matrix through non-negative matrix factorization with a set number of factors, let's say 10, results in 10 groups of neurons. Each of these groups will also have activation values associated with each time step, just like a neuron, where these activations are actually an aggregation over the neurons in that group. Hence, each group can now be analyzed independently to find meaningful patterns, just like we try to analyze individual neurons. Now, since one can set the number of factors to some small value, matrix factorization methods result in groups of neurons that are easy to analyze, because there are fewer of them. However, the number of factors needs to either be pre-decided, or a search needs to be run over various values of this hyperparameter to fully explore the outputs. Another limitation of this method is that the groups are computed per prediction or per input, and changing the input may result in different groups altogether. Hence, one needs to perform some kind of aggregation over a dataset to make broader claims regarding groups of neurons. It is not clear yet how such an aggregation can be performed because matrix factorization is fairly new in the NLP domain, but it is still quite a promising direction. So the next set of methods we will look at is our clustering methods. The assumption in clustering methods is that if a group of neurons targets specific concepts or classes, then their activations would naturally form clusters over some dataset. Authors have looked at clustering from various perspectives. Some have used activations directly, while others have performed clustering on some data derived from these activations. And we will take a look at a couple of these techniques now. The work by Maes et al. Is an example of using the activations directly, where their aim was to cluster the neurons in order to better understand the differences across output classes. One thing to note is that clustering does not work very well in high-dimensional spaces, which is the case when a model has a large number of neurons. Hence, Maes et al. Proposed the idea of using dimensionality reduction techniques like UMAP to first reduce each neuron vector into a lower dimensional space and then perform the clustering. After getting these neuron clusters, they chose to analyze them visually. For example, they were able to identify groups of neurons that activate together for specific output classes in some computer vision models, and this can be seen in the figure on the right. Apart from clustering on raw activations, another proposed method is to cluster neurons based on their redundancy. The assumption behind this method is that many neurons are redundant within a network and activate in a similar fashion over similar inputs. Formally, the correlation between neuron activation pairs is first computed, and the neurons with a high correlation are considered redundant. Let's look at a concrete example. Here we have neuron vectors extracted over a dataset for 4 neurons, and each neuron vector is a row in this table. The Pearson correlation is first computed between each neuron vector pair. In this case, we can see that neurons 1 and 3 have very high correlation as highlighted in green, and neurons 2 and 4 have a very high correlation as shown in orange. The authors use this to show that these neurons pairs are redundant with each other. The final step in their proposed method is to perform clustering over the correlation matrix obtained in the previous step. All neurons falling within the same cluster can then be considered to be redundant, and we can assume that they're learning very similar information. The limitation of this method is that the number of clusters again needs to be predefined, and using a very high number of clusters may lead to redundant neurons being placed in separate clusters. On the other hand, using a very low number of clusters may lead to dissimilar neurons being grouped together. So finding the balance again is key in this method as well. So finally, the last type of method that we will see under the umbrella of unsupervised methods is multi-model search. This type of method uses multiple models that are trained for the same task. The idea is that if multiple models learn the same concept independently, then that concept must be important for the task. Conversely, if some concept is important for a task, then all well-trained models for that task should learn the concept. Hence, the goal of multi-model search is to find neurons across multiple models that behave similarly on a given dataset. The final ranking of neurons within a model is computed based on how similar each neuron is to neurons from all of the other models. So looking at this a little bit more formally, Bao et al. Proposed calculating the Pearson correlation scores for each neuron against all other neurons from all other models. They then aggregate the correlations for each neuron using several different ways with the aim of highlighting different properties and different findings with each aggregation. Specifically, they proposed 4 schemes: max correlation, mean correlation, regression ranking, and SVCCA. At a very, very high level, using max correlation helps to capture properties that emerge strongly into separate models. Mean correlation, on the other hand, selects neurons that are correlated with many other models, even if they are not overall the most correlated ones. Regression ranking finds neurons whose information might be distributed in other models. And in a very similar fashion, SVCCA captures information that may be distributed in other models, but in fewer dimensions than the whole representations. Overall, multimodal methods work quite well, especially when you have several well-trained models to identify patterns that are salient for any given task. So that wraps up our method section. Let us now switch gears and take a look at how we can perform evaluation. Just to remind ourselves, the goal of each of the methods is to give us a sense of important neurons in a model, whether that is importance with respect to a particular concept or to the model itself. And while we have now seen these methods in some detail, an important part of the concept analysis pipeline is evaluation. Essentially, we want to evaluate if the group of selected neurons or a ranked list of neurons correspond to actual importances in the model. More formally, we want to answer the question of how correct the neuron ranking, selection or clusters from any given model is. So let us take a look at some of these evaluation methods, starting with ablation. Recall that we have already seen ablation as a technique for identifying important neurons. In evaluation, we use ablation from a different perspective. In this context, the question that ablation tries to answer is whether a given ranking actually reflects the importances with respect to the original model. The actual technical implementation of ablation is still the same. We still clamp the output values of the neurons to be ablated. So Bao et al. Use this, and they have performed this evaluation for machine translation models by ablating some percentage of top and bottom neurons in a model. The assumption here is that if the ranked list is correct, then the drop in performance with top neuron ablation should be much higher than with bottom neuron ablation. So for example, in this figure, we can see that when we increasingly ablate top neurons represented by the blue line, the drop in performance is immediately steep as compared to the orange line. So a side effect of this is also that if a ranked list is with respect to some concept rather than the overall model, then ablation will reflect the relative importance of that concept to the model. Dalvi et al. Performed a similar evaluation with language models. Notice that the curves are actually inverted here, as the metric that is being used to measure performance is perplexity. And the higher the perplexity, the worse the model. Hence, it is important to keep in mind what metric is being used so that the performance curves are interpreted correctly. We can take ablation one step further and also use an external classifier's performance to perform a similar evaluation of the input ranking. Specifically, instead of ablating neurons in the original model, we can use the top and bottom neurons from any model and train an external classifier using the activations of just these neurons. This is very similar to neuron probing, where we use the activations and supervised data to build a classifier. Similar to ablation, we look at the performance using just top or bottom neurons to evaluate the input ranking. In this case, the classifier trained with top neurons should have a higher accuracy than one trained with bottom neurons. Using an external classifier rather than ablation enables us to evaluate the input ranking directly against a concept instead of looking at the overall model, as we are using the performance of a model trained specifically on a single concept's data. So various authors have used random neurons as well, both here and in ablation, to further compare the top and bottom neurons. The next evaluation technique we will look at is cluster comparison. This is specifically used in the case of some unsupervised methods where we only get clusters of neurons rather than an explicit ranking. The usual assumption is that neurons within a cluster represent similar knowledge. And so we can use ground truth clusters if they are available and compare them to the clusters generated by some method. Hennigan et al. And Michael et al. Have used several techniques in this case, starting with standard clustering evaluation metrics like B cubed, which look at precision and recall of each neuron's predicted cluster with respect to goal clusters. They also tried training external models with neurons just from a single cluster to evaluate against particular concepts. And finally, Michael et al. Also used several other metrics like diversity and uncertainty that take a look at the general makeup and composition of the clusters themselves. And this enables to evaluate the clusters in a more qualitative fashion. And finally, let's look at the general class of qualitative evaluation methods. We have actually already seen several of these techniques like visualization and corpus selection that were used to manually identify important neurons. So over there, we started with all neurons in the network and shortlisted them based on these methods. In the case of evaluation, we already have a ranking, or we already have a small subset of selected neurons. And so what we can do is then visualize or perform corpus selection specifically on these selected or top neurons. This is much more feasible in this case because the number of neurons of interest has been significantly reduced to a manageable set. Now, despite the fact that a number of these evaluation methods have been proposed and applied, there are still some major caveats in this area that are required to properly evaluate the efficacy of these methods. Currently, it is difficult to compare results across different models. People use different data sets, and it is not clear if the identified neurons reflect the behavior of the model itself or of the data set. To overcome such problems and many other standard evaluation benchmarks in interpretation are required, where researchers are able to conduct studies and directly compare results with previous work to establish the efficacy of new methods. Moreover, due to the very complex nature of models today, various neuron and neuron groups may be present for the same concept. Two methods may choose to exploit this redundancy in different ways and select very different neurons as important neurons. In this case, both rankings or selection may actually be correct, but it is not clear how this can be evaluated robustly. And finally, many concepts may just not be interpretable in terms of natural concepts, especially those derived via unsupervised methods. Having a human in the loop in this case to perform manual analysis of these neurons and these patterns may be quite helpful in case these automatic methods fail. So that is all for evaluation, and I'll hand over to Nadir now to discuss the many findings and analyses from these methods that we have seen today.
Alright, until this point we have discussed the models to discover neurons in the network and approaches to evaluate the efficacy of these methods. Let us now talk about the findings that came out of different methods and what analyses were carried out on top of these findings. We categorize these into three classes, where we discuss what concepts were captured within the neurons, how the information is distributed across the network, and how does it compare across various architectures. People often ask, what are any applications to neural interpretations beyond understanding the mechanics of the neural networks? So we will also touch upon some applications such as efficient feature selection, manipulation where the system's output is changed through relevant neurons. Some other applications that we are looking at are domain adaptation and causal explanations. So without further ado, let's dive into the findings and see what neurons have been discovered. In the concept discovery section, we discuss the work that analyzes neurons corresponding to human interpretable concepts. For example, lexical concepts such as morpheme, words or phrases, or linguistic concepts such as part of speech tags or semantic tags. One of the earliest work done on analyzing and interpreting neural NLP models was done by Karpathy et al., who found some really interesting language properties captured by the neurons. For example, here is showing a neuron that is capturing position of a word in the sentence. It activates positively at the beginning, becomes neutral in the middle, and then starts activating negatively towards the end. Another interesting neuron that they found captures long distance dependency. So whenever there is a quotation, the neuron activates unless a matching quote is found in the input, at which point it stops activating. Other papers also demonstrated specialized neurons to capture long-range dependency. For example, Lacratz et al. Found memory cells in LSTM models that capture subject-verb agreement. As you can see, the cell is active when the subject appears and remains active until the agreeing verb arrives in the input sentence. In a similar vein, Lee et al. Discovered very interesting intensification neurons in the LSTM models trained towards the task of sentiment classification. So whenever modifiers like a lot or so much appear, then these neurons become active to change the sentiment of the sentence or intensify it. So I like it a lot. So you are intensifying the sentiment. Similarly, they showed that some neurons learn a negation phenomena. So whenever a not is added to an adjective or an adverb, it negates its effect. Although the pattern for negation were not as clear as the intensification neurons. They also found an asymmetry in the behavior of the negation neurons, where negation plus positive words were clustered with the negative words, but negation plus bad wasn't showing the same behavior. So this demonstrates that a neural model is able to capture subtle differences in the composition of different words. Similar findings were shown in the LSTM-based neural machine translation models. For example, Dalvi et al. Analyzed the encoder representations with respect to different morphological and semantic properties and found many neurons corresponding to verbs, article, location, and name entities. Here we are showing that it captures present and past tense verbs in English and articles in the German encoder. NAACL et al. Studied convolution neural networks trained across different tasks and datasets and found neurons corresponding to specific natural language concepts such as very shallow lexical concepts like words or phrases or morphemes, or auxiliary verbs like should, would, not, can. So some of the neurons were capturing larger chunks of words like preposition phrases on the left of the screen and negative and positive adjectival phrases when the model was trained towards the task of sentiment classification. Rather than trying to discover the neurons that capture specific linguistic or predefined phenomena, some authors also tried to analyze what most important neurons in the network do. So they did a cross-model correlation analysis to find out what is the most salient information in the network, what are the most important neurons. So they found that position neurons that fire based on whether a word is at the beginning or middle or end of the sentence is to be the most important neuron for the task of machine translation. And they found it consistently across different language pairs and different training runs. Other neurons that were most important were parentheses and punctuation and conjunction neurons. We also saw this position neuron earlier when we were discussing Karpathy et al.'s work. They also found this neuron in LSTM based language model. So maybe these are not necessarily MT related neurons but very important language neurons. One interesting neuron that authors found was a switch neuron, which activates positively for one phenomena and negatively for another phenomena. So here you can see that this neuron is activating positively for the present tense verb and negatively for the past tense verb. These are very interesting neurons that we will see later that they're really helpful in manipulating systems output. So please remember this and we'll come back to this later. Sometimes salient neurons in the model correspond to architectural details. For example, Lee et al. Found two most salient neurons obtained in the glove representations were actually the frequency neurons. And on the task of part of speech tagging, semantic role labeling, or name entity recognition, these neurons were consistently found to be playing an important role. Another unsupervised method that decomposes activation metrics into manageable factors that we discussed is the NMF model. The authors show how various neurons combine into factors towards each generated token in GPT model. Using 10 factors, we can see several informative group of neurons with respect to a concept. For example, Here is a factor that is focused on the comma words only. A factor that is learning functional words such as preposition or articles. A factor that is capturing capitalization. However, in this example, you can only see one word, NAACL, that is capital. We think this is actually a capitalization factor. Similarly, a factor that is highlighting the year in this text. Corpus-based methods analyze neurons by looking at their top K activation contexts. Here the authors found neurons related to very specific concepts in multimodal image captioning tasks. For example, they found neurons related to electronic items. As you can see in the trigram context, this includes laptops, cables, and cords. They also found neurons that were capturing a group of people concept. So you could see group of men, crowd, team, etc. in the context. Finally, a neuron that's focusing on the salad items. Another work done along this line is that rather than extracting contextual information from the corpus, we generate the context that maximally activates the underlying neuron. So this method brings out hidden information that network has but the underlying corpus cannot reveal for you. For example, the original corpus method found that neuron 522 is actually a horse neuron. However, when you generated additional context for this neuron, you can see that it activates for motorcycles as well, suggesting that it's a general racing neuron and not just limited to horse racing as we speculated earlier. So this shows that additional piece of information can surface via this method, and it is potentially helpful to strengthen the interpretation. Work done on representation analysis has shown that cold linguistic tasks such as morphology or syntax are captured within deep neural networks. Dalvi et al. Carried a more fine-grained analysis and discovered neurons related to each class within these tasks. For example, neurons related to nouns, adverbs, adjectives in part of speech tagging, or neurons related to location or events in the semantic tagging. In their analysis, they found that some concepts such as functional words are localized to fewer neurons, as opposed to open class words that are distributed to many neurons. So for example, month of the year and WH adverbs require fewer neurons in the network as opposed to nouns and adjectives, which are open class categories and are captured by many neurons. This observation was consistently found to be true across different languages of varying morphological granularity, and was also confirmed in the pre-trained transformer models recently by another work done in this direction. Alright, up till now we have seen how the discovered neurons align with human interpretable concepts. Now I will go through some of the architectural analysis that were carried to, for example, understand how the information is distributed within the network, how redundant it is, and how it varies across different architectures. Such an analysis helps us to understand how the information is stored and propagated in the network. It also allows us to compare different architectures. Representation analysis showed that core linguistic properties are captured within deep neural networks. A recent work on analyzing neurons in pre-trained transformer models discovered that a minimal set of neurons can be extracted to perform different linguistic tasks. The authors showed that lower-level tasks such as capturing word morphology or lexical semantics require fewer number of neurons as opposed to complex CCG supertagging tasks. So CCG is kind of a task that is deemed equivalent to parsing, and the authors showed that it requires up to 15% of the network to capture such complex phenomena. Once they found the minimal set of neurons required to carry different tasks, they analyzed how the spread of neurons is across the network and across different layers in the network. There's a lot going on this slide, so there are 4 tasks: part of speech, tagging, semantic tagging, chunking, CCG tagging, and we study the spread of neurons in these tasks across different layers in three architectures. If I break it down for you, the results on the left side show that lower level tasks such as word morphology and lexical semantics are captured at the lower layers. So the neurons that were found were predominantly appearing in the lower layers of the network. On the other hand, if we look at the right side, where you are seeing the complex syntax level tasks, final layers seem to be contributing more towards the salient neurons. In another research on interpreting pre-trained transformer models, Shaw et al. Analyzed submodules within a transformer block and showed that shallow layers in a transformer block accumulate more concepts than deeper layers. The task here is word sense disambiguation or homograph, where a word could mean different things based on contextual information. And they found this to be consistently true across all layers of the GPT and Roberta model, where you can see that expanding layers are more prone to learning concepts as opposed to the projection layers. They also found that concepts that learn homographs are much more spread across the network as opposed to sense neurons that were predominantly found in the lower layers. The spread of neurons allows us to compare different architectures. Here the authors found that although BERT and other transformer models require roughly the same number of overall neurons to carry different linguistic tasks, the neurons are spread differently across the layers. For example, the information in BERT was found to be more distributed than XNet, where it seemed to be localized to specialized layer. Looking at the XNet, it is very clear that morphology was learned at the lower layers and syntax at the final layers. This is not evident in the case of BERT, where the information seems to be distributed, although it still shows that more dominant neurons for CCG are in the higher layers. Shaw et al. Compared Roberta and GPT, one of which is an autoencoder model and another is the generative model, and found that the neurons that were found for the tasks are distributed differently. Another comparison point if the neurons are localized or distributed to many concepts. So here the author showed that BERT has more polysemous neurons that are attending to many concepts as opposed to XNet, where the neurons were more exclusive to a particular concept. Such a finding can be potentially useful when we try to control networks' behavior by tweaking these neurons. It is easier and cleaner to manipulate a system when we can identify neurons that are related to one or two concepts as opposed to manipulating neurons that are attending to many unrelated concepts. Another piece of work that uses minimal neurons to draw a comparison between different representations is from Henning et al. So they compared representations from BERT model and FastTech, which is a static embedding model using morphosyntactic concepts. And they saw that static embeddings required fewer neurons to capture the same information as opposed to contextualized representations where the knowledge is distributed in many neurons. So in BERT they found that 35 neurons were required to obtain the same performance as opposed to static embedding where they required only 2. Similarly, Shaw et al. Discovered that more specialized neurons are found in Roberta and GPT representations as opposed to BERT and variants. This resonates with the finding that we just discussed a few slides ago, where we found neurons in BERT to be more polysemous as opposed to XNet. So these researchers confirmed the same result that by seeing that more expert units were found in Roberta and GPT as opposed to BERT, where the expert units are fewer and more neurons are polysemous. We were talking about minimal set of neurons or expert units that can be aligned with the concept, but since neural networks are innately distributed in nature, this causes redundancy in the network. So some researchers carried a redundancy analysis. They identified most salient neurons in the network that carry the task, but then also found other neurons that can be used as a fallback option when we remove the most salient neurons. So the overall network performance still remains in this intact when you remove the most salient neurons. Dalvi et al. Showed that a significant amount of redundancy exists in these networks. They saw that if you remove 85% of the neurons in BERT or 92% in XLNet, it still maintains a decent amount of accuracy on several downstream tasks. And they made use of this observation in efficient feature learning, which we will be discussing later. Similarly, Shin et al. Found neurons exhibiting a variety of roles, where some neurons were found to be exclusive to single concepts, some captured many concepts like BERT where they were polysemous, and neurons that are not sensitive to any concept in the network. They also found that while there are specialized neurons in the network, removing them caused deterioration in specific class accuracy, but overall network performance does not deteriorate, showing that the information is redundantly distributed in the network. A similar observation was made by Lee et al., where they observed that removing specialized frequency neurons, which we saw earlier in the glove model, that is responsible for predicting most of the properties in many tasks, if you remove these neurons, the network was still able to obtain the same accuracy. They also studied how dropout affects this and found that training networks without dropout, they were able to find more specialized neurons as opposed to when the networks were trained with dropouts, as we know that dropout causes the information to be distributed and therefore redundancy appears. Right then, so moving on to the last leg of this section, we have seen how identifying salient neurons in the network and how they align with human interpretable concepts can be helpful in understanding the black box. But there have also been numerous potential applications that it can be put use to. For example, it can be used to control networks behavior. People have shown that it can be used for efficient feature selection. It can be used for task-specific model compression, where you can remove irrelevant or unimportant neurons with respect to a task and have a distilled model. Some folks have used it for domain adaptation. It can be used for guiding architecture search. Now I will discuss some of the work that has been carried in this direction. So once you have identified the neurons that is related to any concept, the most forthcoming application is being able to control these neurons to be able to change system behavior with respect to that concept. For example, if you want to control the output of machine translation system with respect to a gender and make it output masculine versus feminine gender of a word, then identifying neurons that control the gender could be useful. Or being able to control the tense verb would allow you to output a sentence in present or past tense form. So for example, the model predicts present tense verb in this context with a high probability, but you want to change the system to output a past tense form of the verb. Perhaps we can find a neuron that controls this phenomena and manipulate to achieve this desired effect. Do you remember the switch neuron that I showed you earlier, which activates positively for the present tense verbs and negatively for the past tense verb? Bao et al. Showed that by manipulating the activations of such a neuron, we can generate present or past tense output in the LSTM-based machine translation system. And they tried it for many different language pairs where they were finding these English verb neurons on the encoder side, but manipulating them, they were able to control the output of the machine translation decoder. They were also able to manipulate gender and number neurons to output masculine versus feminine variant of a word or singular versus plural variant of a word respectively. Although here they were not as successful as with the case of verb neurons, where they saw that 67% of the times they were successful. Here the accuracy was a bit on the lower side. Another notable attempt to manipulate systems behavior is from Sauertal, who induced topic level concepts like bird and lead and showed that in text generation, by identifying neurons that are related to these concepts, they can manipulate the system to start generating text around these concepts. Both of these works were pilot studies and they used manipulation as a proof of concept, but they open very interesting research questions. For example, neurons capture concept information redundantly, as many papers have shown. Manipulating a single neuron or a single group of neurons, the network may fall back to use other neurons designated for that job. It is non-trivial what group of neurons that were aligned to a concept actually take part in manipulating the output. Also, different subset of neurons may play a role in different examples. How do we find which neurons are responsible for manipulating a particular set of examples? How do we find optimal value for a neuron at which it can be effectively toggled to manipulate the output? How do we deal with polysemous neurons? They play varied role in the network. Also, neurons are so interconnected and manipulating them may cause undesirable effects on the other neurons in the network, thus resulting in unstable network performance. All these questions, along with many other questions that would pop up when someone takes on this task, require answers. So this is a very interesting research frontier to push forward to. An application that directly stems from redundancy analysis is efficient feature-based transfer learning. Feature-based transfer learning has shown to be a viable alternative to fine-tuning approach which is commonly used. Dalvi et al. Devised an efficient feature-based learning algorithm that exploits various types of redundancy present in the network. They first target layer-wise task-specific redundancy using layer probes and see which layers were redundant. Then reduce the number of layers required in the forward pass to extract contextual representations from only part of the network. Then they filter out general redundant neurons in the contextualized embedding using correlation clustering. Lastly, they use task-specific redundant neurons using linguistic correlation analysis to find neurons that were only responsible for that particular task. Doing this, they were able to reduce the neuron set to less than 100 neurons for several tasks while maintaining more than 97% of the performance of the oracle that uses the entire network. The procedure achieves a speed up of around 6.2% in computation time for sequence labeling tasks as opposed to using all the features. Another application that I am going to discuss is domain adaptation via neuron pruning. The typical fine-tuning method where you train a general model on a lot of data and then fine-tune it towards in-domain data is prone to catastrophic forgetting of the general domain. The idea here is to identify the most salient neurons for the translation model and preserve them during fine-tuning process by using only the less important neurons during the fine-tuning process. In the first step, they rank neurons based on their importance using Taylor expansion algorithm where they directly approximate the change in the loss function when removing particular neuron. In the second step, they remove unimportant neurons from the network and try to restore the loss in performance via student-teacher model, where the full model is a teacher model and the pruned model acts as a student. In the final step, they expand the pruned network to its original size and fine-tune the model towards in-domain, but only using the unimportant neurons that were filtered during the first step. They freeze the part of the network that they preserved for the general domain, and using this approach, they help avoid catastrophic forgetting of the general domain, while also obtaining an optimal in-domain model. The findings that we saw earlier use visualization to explain the function of a neuron and align them with the concept. Another piece of work that was recently published by Mu et al. Used a different technique where they build explanations using composition of logical operators. For example, in an NLP task, this neuron 870's logical expression looks for a specific pattern like absence of a man in hypothesis and presence in the premise, or a mention of eating in the hypothesis and absence of woman in the premise, or mention of dancing in the hypothesis. So quite a vivid description, but this description maximizes the role of this neuron in the task in terms of intersection over union, a metric that the authors used. The authors also used these shallow concepts such as presence or absence of a word to create adversarial input that they showed could change the prediction inaccurately. For example, they found a neuron that solely relied on the presence of nobody in the hypothesis and were able to spuriously introduce this word in the input to change the prediction. The authors also tried to connect interpretation and model performance. In the vision domain, they found that generated explanations to positively correlate with the prediction of the model, but they did not find this to be true in the NLP task. The authors conjectured that this could be due to their analysis on shallow word level concepts in the NLP task. If shallow concepts such as word presence are highly interpretable, this may point to the brittleness of a given model. Identifying the right concepts for the task may lead to a more positively correlating trend. Another piece of work that tries to measure the importance of neurons with respect to actual models' performance is via ablating neurons related to that concept. The idea here is to evaluate how important each concept or set of concepts is to the end model. So the blue curve shows the most salient neurons. As you can see that when you ablate just 100 neurons, it causes a degrade in 20 blue points. The authors here were trying to study how important are the morphological or semantic concepts. So if you remove the morphology related neurons or semantic neurons, would you see a similar drop in performance? And that way we can confirm how important or how overlapping these neurons are with respect to the most salient neurons of the model. And what they found was that although morphology and semantic neurons are important for the task, there are other phenomena which are critical for models' prediction, such as sentence length and punctuation neurons. With this, we would like to end this section and move on to the next section.
Thank you, Nadir, for sharing these findings and results. Hopefully, all of you now have a better idea of the work that has been done in this field. We are excited to see this field grow and hope to spark some ideas by showing these findings to you all. So now, let us look at some concept analysis from a more practical perspective. We will first briefly touch upon datasets before looking at actual code and toolkits. There are many things that one needs to keep in mind when choosing the dataset for neuron analysis. Most work today looks at word annotated data, since individual token activations are somewhat easier to analyze compared to aggregated sentence summary vectors. One also needs to remember that not all techniques we have seen today will work equally well on the same dataset. The granularity at which a dataset is annotated is also very important. For example, one can either have a single high-level category like verbs, or subdivide the data into finer categories like past tense verbs and present tense verbs. It is also important to have a human in the loop, especially when starting out with the analysis, to see if the neurons that are being extracted are actually meaningful and mean something to an expert. Finally, some authors have considered automatically generating datasets to overcome the limitations of not having annotated corpora. And they have looked at several things like extracting knowledge from past trees or even datasets or databases like WordNet and SenseNet. Just as a reference, here is a non-exhaustive list of datasets used in the papers we have discussed today. So now let's move on and take a look at some actual toolkits and code that we can use to perform concept analysis. This field is still relatively new, so not a lot of toolkits that target neurons directly are available. We will discuss 3 toolkits today that can help us in visualizing individual neurons or groups of neurons, as well as help us in analyzing the role and functions of these neurons. The first toolkit we will discuss is LSTM-Viz. This toolkit provides a very nice GUI to analyze individual hidden states in LSTM models across sentences, as well as find neurons similar to a given neuron in its activation patterns. The toolkit also comes with some pre-prepared data associated with linguistic properties like nesting and phrase structure, and this can be quite useful in analyzing models from a linguistic perspective. Their source code is available to download and use locally. The next tool we will look at is Echo. This is a fairly new tool by J. Alamar, who many of you will know from the illustrated transformer. This tool encompasses a lot of features, some of which include visualizing activations of particular neurons, performing saliency analysis for every token interactively in a browser, and even finding groups of neurons working together using non-negative matrix factorization. Several Google Colab notebooks have also been provided with sample interaction analyses, and the source code for the toolkit itself is available on GitHub, and it's also provided as a pip module so that it can be easily integrated into your own code bases. The final toolkit we'll look at, and we'll go a little bit more deeper in this, is NeuroX, which has been released by our group here at QCRI. NeuroX is a neuron analysis focused package that implements several techniques from the ones we have seen today. It also provides easy-to-use extraction modules for all transformer models, with support for other libraries coming very soon. The entirety of neuron probing and correlation clustering that we have seen today can also be done using various methods provided in this toolkit. Let's take a closer look at some of the methods that we can use and process some actual toy data through the entire pipeline. All of this data, as well as a notebook with all the code you see, can be found in the examples directory of the repository, or you can also directly go to this link. Before we begin any kind of neuron analysis, we first need to have some neuron activations from some model on some given text. In this walkthrough, we'll be considering a small data set of 10 sentences with the following sentences. Extraction is as simple as importing the correct extractor for your model. So for example, if our model is a transformers model, let's say BERT, we would use the transformers _ extractor module. We can then pass in the name of the actual model. So as I said, in this case, we will extract activations from a base BERT model, the uncased version. And extracting using any other transformer model simply requires passing in the correct name into this function. We also pass in the input text as well as where we would like to save the final activations. Take a look at the API documentation for all the options provided by this function, including various kinds of aggregation over subwords, various output file types other than JSON, and so on. Once this function finishes running, the neuron activations will be saved inside activations.json. The next step in performing neuron probing is to prepare some annotated data. In our toy example, we have annotated each word with its part of speech tag. So in this case, DT is determiner, NN is noun, and VB, all the VBs are verbs, and so on. In real life, you would either choose a human annotated data set that's already available, or try to generate one automatically, as we discussed earlier. Once we have our supervised data ready, let us first load the extracted activations from the save file. Remember that the actual extraction takes place only once because it's an expensive process, and then from then on we always just load the activations. The built-in data loader module has all the functions necessary to load activations and the supervised data. So here we can see that the activations are just a bunch of numbers after we have used the load _ activations function. And here each number corresponds to a neuron's activation for a particular token. The next step is to load our supervised data, the actual sentences and the part of speech tags. The load data function can help us do this. We pass in the source tokens, the target labels, as well as the activations. Apart from loading the data itself, this function also ensures that all of these three things are parallel. That means is every token in the source should have an associated label as well as a set of neuron activations. Any sentences that do not match this are discarded to a certain limit. So you can be sure that the data returned by this function is going to be clean and parallel. Now that we have all the data loaded, we can start doing our neuron probing. The first step in this process is to convert the data into a form that can be fed into the training, evaluation, and analysis pipeline. This can easily be done using the create _ tensors method provided in the library. that takes the loaded tokens and activations and converts them into standard shaped tensors of numbers that can be directly used downstream. A mapping dictionary is also returned so that the user can map the numbers back to actual words and class labels. The API reference has all the details and also has more options that you can use when calling this function. We can now finally train our probe using this process data. A logistic regression probe can be used for classification tasks, which is the case in our toy example, but a linear regression probe is also provided for regression tasks. The train function also accepts a lot of training parameters which can be found in the API reference. Here by default we are just training using some regularization parameters and for 10 epochs. Finally, this function returns the actual trained probe back. Recall that we discussed that after training a probe, the first thing to do is to actually evaluate it. So the toolkit also provides methods to evaluate a trained probe using a wide variety of metrics. It returns not only the overall score of the probe, but also class-wise scores to see if the probe is doing better on certain classes. Once we have looked at these scores and we are confident in our probe, we can use various methods to extract set of neurons from our trained probe, such as top neurons, bottom neurons, or random neurons. An example is shown here for getting the top 1% neurons globally across all tags, as well as the top neurons per class. Similar function calls can be made for bottom and random neurons. The ablation submodule can then be used on these extracted neurons to further evaluate our probe and selection. For example, we can mask all non-top neurons and evaluate our probe, and the accuracy should remain relatively high since we are only making use of top neurons. The example method shown here is a masking method, but other methods are also provided that completely remove certain neurons to evaluate those sets more independently. Apart from getting sets of top or bottom neurons, the toolkit also provides a way to get a global ordering of all neurons. Hence, the neurons here the neurons are ranked by their importance to all classes in the given tag set. So, for example, we have printed the top 10 global neurons across all part of speech tags in our toy dataset in this code snippet. It is also common practice to train probes only on certain subsets of neurons, whether that is to look at the overall performance of the probe using those neurons or to analyze specific modules in the original model. For example, one can use some of the helper methods to filter activations and keep the data for certain layers only. Here, we are filtering activations just from layer 0 and training a probe on layer 0 alone. This can be very useful to compare performances and neuron distributions across layers to see how the representations change as we move deeper into a network. Finally, the toolkit also has some helper methods to visualize specific neurons in specific sentences. As we have discussed several times today, having a human in the loop is very important in many circumstances. Being able to directly visualize, say, the top neurons across a dataset can be quite insightful for researchers. And we can attest to this because our neuron analysis journey began exactly this way. So before I close this section, I would like to point out a couple results that have been produced using this toolkit so that the connection between the code and theory we have seen today is a bit more clear. We have seen that the get top neurons method can be used to extract top neurons for every class. This was used to show that open class concepts like nouns and adjectives usually require a large number of neurons, while closed class concepts like WH verbs and month of year can be effectively encoded in just a few neurons. The ablation module has also been used to perform layer-wise analysis. The top neurons were used, again in this case, to see how the knowledge is encoded across layers in a pre-trained language model, with some models having more or less equal neurons across different layers, while others having very few layers with a high number of neurons. So that's all for this toolkit and also the practical section. You can visit these links to access the source code, use the toolkit, and read the API reference. A GUI is also available that uses the toolkit underneath and allows you to explore neurons interactively. An updated version of the GUI is coming soon with more fixes and support for more libraries. So this brings us to a close and to the end of the first high-level section of the tutorial, which was concept analysis. Let us now move on to attribution analysis. Recall that all methods we have seen so far identify neurons with respect to a concept, but they do not tell us if these neurons or concepts were ever used in any prediction. Attribution analysis answers this exact question, and I will now hand it over to Narine to talk about this.
In this part of the tutorial, we will focus on the causation analysis using approaches that will help us to estimate the importance of different neurons for a specific model prediction. This type of approaches are also known as attribution algorithms. Let's assume that we are given a neural network that takes an input features and produces an output prediction target class index between 0 and p. Let's denote neural network by a function fc where c is the prediction class. Let's say that we want to know how important the neurons are in layer L for predicting target class k given a set of input examples. It is important to note that with neurons in layer L, we refer to the activations in layer L, as also introduced in the previous sections of this tutorial. In order to access the neurons in layer L, we need to break down fc function into two components, fl and hlk. fl will return us the activations in layer L, and hlk takes those neuron activations from layer L and produces the final prediction score. Our goal is to understand how important the neurons are in the layer L when predicting target class K. There are a number of attribution algorithms that will allow us to compute neuron importances for layer L. One of the most simplistic attribution methods is saliency. Saliency is a gradient-based attribution algorithm that helps us to compute the importance of specific prediction with respect to layer neurons. Formally, it explains the changes in the output prediction on the basis of infinitesimal neuron activation perturbations and is formalized as the gradients of output prediction Fc with the neurons in layer L. Some of the shortcomings of saliency method are that gradients can be noisy and sensitive to functions input and unfaithful to functions output. You can convince yourself by trying out simple neural network function and compute the gradients and see how noisy they can become. A technique called smoothgrad helps us to mitigate the issue of noisy gradients by adding Gaussian noise with a standard deviation sigma to input examples and compute the expected value of explanations in the neighborhood of input x. In other words, this technique helps us to perform smoothing by approximating with Gaussian kernels. There are also a couple other variants of this technique called vargrad or smoothgrad square that you can check out in the original paper. Although smoothgrad helps us to mitigate some of the issues with noisy gradients, it also has several limitations. For instance, proposing optimal number of examples n can be challenging. Also, if n is very large, it can be computationally expensive, and in that case, we need to think about distributing our computations across multiple GPUs or other computational resources. Integrated gradients is another technique that can be used to compute neuron attribution or neuron importance built on top of gradients that integrates the gradients along a straight line from baseline x0 to input x. Baseline x0 oftentimes represents an uninformative input, which is compared and contrasted with the original input x in order to draw conclusions about the features that stand out or are more important in the original input x. In case of images, black image, white image, or a noise, a blurred image can be example of a baseline. In case of text, a baseline could be a padding token or any other uninformative token that we can think of. The baseline for the activations will be the output of FL function for the inputs x0. In order to compute an integral along the straight line from inputs from baseline to inputs, we use alpha scaling factor, which helps us to choose a point between the baseline and the input. In this case, since we want to understand the importance of neurons, we compute the gradients with respect to the output of the layer L. We then use Riemann sum or Gauss-Lagrange to approximate the integral. Formally, the attribution phi for the neurons in layer L is the product of layer activation differences at its inputs and baseline and the integral of gradients of neurons in layer L along the straight line from baseline to input. Some of the limitations of integrated gradients is that finding good baselines can be challenging, and integrated gradients does not account for feature interactions and correlations. Integrated gradients satisfies a number of axioms. One of them is the axiom of completeness, which states that the sum of the attributions is equal to the differences of a neural network function's output at its input and baseline. This axiom is closely related to the fundamental theorem of calculus for path integrals. The second axiom is the axiom of sensitivity, which states that if the input and the baseline differ at one feature and result to different prediction scores, then for that different feature we will get non-zero attribution. The third axiom is implementation invariance, which states that the attributions for two functionally equivalent neural networks are identical. Layer conductance extends integrated gradients using chain rule with respect to internal neurons. The difference between integrated gradients and layer conductance is that instead of computing the integral of gradients of model output with respect to the neurons in layer L, layer conductance computes the gradients of model output with respect to the internal neuron layer L, and then multiplies it by the gradients of internal layer neurons with respect to the model inputs. The rest of the formulation is similar to integrated gradients. Similar to integrated gradients, layer conductance also satisfies the axioms described in the previous slides. One of the most simplistic perturbation-based approaches is neuron ablation. Similar to gradient-based methods, we want to understand how important are internal neurons in a given layer L for our prediction target K. Neuron ablation measures the importance of neurons based on the magnitude changes in prediction scores or magnitudes of prediction goodness, such as loss or accuracy, when the neurons are ablated. Let's look deeper what this means and how we ablate the neurons. Let's assume that in this example, the neurons are represented as a two-dimensional matrix where y-axis represents the number of examples and the x-axis is the number of neurons. In order to ablate neurons, we also define neuron ablation masks, which have the same shape as our inputs or have the same shape as one of the examples in our input. Neuron ablation masks help us to define groups of neurons that we want to ablate together. As shown on this slide, we want to ablate first and third neurons together. Therefore, those two neurons have the same mask ID. Besides ablation masks, we also define the ablation values, also known as baselines, that are used to replace the original neuron values. Traditionally, zeros are used as ablation values, but we can also use customized default values. We recall HLK function from previous slides that helps us to estimate the output of model given layer activations or neurons as input. Neuron importance in this case represents the magnitude changes of a differences in the function's output before and after neuron ablation. Before ablation, HLK function takes the output of FL function as inputs, and when we want to perform ablation, we pass to HLK function the output of FL functions with some neurons being ablated. Some of the shortcomings of neuron ablation are identifying which neurons to ablate together and what values to ablate with. Besides that, the ablated neurons might lead to model state, which wasn't observed during the train or validation phases of the network and could lead to predictions with high uncertainty scores. In addition to that, neuron ablation methods can be computationally more expensive compared to gradient-based methods, depending on the number of neurons that we have in our internal layer, or depending on how we group those neurons in our internal layer. In this section of the presentation, we will focus on feature interaction and examine feature importance from the perspective of feature interactions. The examples are based on input features, but they can also be applied to internal neurons in order to understand different interaction patterns between neurons. The consideration of feature interactions comes into the picture when the attribution of a phrase isn't equal to the attribution of its constituent tokens. In general terms, we say that an interaction is present when the whole is different than the sum of its parts. For example, in this case, the attribution score for not interesting phrase is not equal to the disjoint importance scores for the term not and interesting. Integrated Hessians is an extension of integrated gradients and is one of the latest approaches on feature interaction detection and attribution, among many other techniques. Integrated Hessians helps us to estimate pairwise feature interactions using second-order derivatives. With the first-order derivative, we compute feature importances, and with the second-order derivatives, we estimate the importance of the ith feature for the importance of another jth feature. For example, on the right side here, we can see that quiet compelling expression, the pairwise interaction of quiet and compelling, has a high attribution score, whereas the interaction score between compelling and S, or intense and steady, is a little bit smaller compared to quiet compelling. Another axiomatic approach on feature interaction attributions is Shapley-Taylor index. Shapley-Taylor index is a generalization of Shapley values for feature interactions that uses higher-order Taylor series terms. It can be computationally expensive depending on how exact we want to be in our computations. Agglomerative contextual decomposition, ACD, is different from previous two methods with the way that it is not axiomatic, but it still returns meaningful interaction attribution scores. ACD clusters the features hierarchically and assigns an attribution score to each group. It is based on the additive decomposition of a deep neural network and has custom adjustments for different types of activations, such as max pool or convolution. On the right side, we can see that although the term very good has a positive attribution score, it conveys positive sentiment. When we put not in front of very good, then its sentiment changes completely, and it becomes negative. So the attribution of not very good cannot be made from the attribution of individual tokens such as not, very, and good. Archipelago is another axiomatic approach, similar to integrated gradients and Shapley-Taylor index. It consists of two stages. In the first stage, it estimates feature interactions, and it can also work on higher order feature interactions, not only pairwise feature interactions. After it detects the interactions in the second phase, it computes the attribution by taking into account the interaction terms that were estimated in the first stage. In this case, we can see some results that were returned from Archipelago. For example, not extreme enough expression has a high interaction score according to this method. In this section of the tutorial, let's talk a little bit about the evaluation and the goodness of interpretability. In other words, how can we know if our feature attribution or neuron attribution methods give us reliable explanations? The formulations that we will have in the next slides are for input features, but they can also be easily extended to intermediate neurons of any model. As we can imagine, the evaluation of attribution methods is challenging, and there is no clear guidance on how to quantify the quality of attributions. Although there are new metrics coming up every day and new sanity check techniques have been developed, most of them are domain specific or are based on specific properties of an algorithm, but it's hard to find a method that is generic and applies to all attribution algorithms. Many people use visual quality checks. However, this can be misleading, and especially in the vision, this can be seen as a confirmation bias. Now let's talk about some quantitative evaluation metrics. Sensitivity max is one of those metrics. Sensitivity max is a metric that estimates the quality of explanations in terms of saddle input perturbations using Monte Carlo sampling-based approximation. Given an input x, its perturbation y on the radius r, a neural network function f, and explanation function phi, sensitivity max is defined as the maximum distance between the attributions with and without perturbations, normalized by the norm of the explanations. Lower sensitivity score means that the explanations are more robust and less sensitive to saddle input perturbations on the radius r. Also, note that although this equation and formulation is for input features, we could extend it and also apply for internal neurons. So similar to sensitivity max, infidelity is another metric that measures the quality of attributions. It is a generalization of the completeness axiom, which we already got to know from integrated gradient algorithm. So given an input x, a neural network function f, a meaningful perturbation I with a probability measure mu I, infidelity is defined as a mean squared error between the dot product of input perturbations and attributions and the differences of our predictor functions at its input and perturbed input. Ideally, we would like to have lower infidelity score. The lower infidelity score tells us that our explanations are more trustworthy. There are other domain-specific metrics for computer vision or for NLP that I will encourage you to check out. In this part of the tutorial, we'll focus on generic open source tools and libraries which offer attribution algorithms and evaluation metrics that we discussed in previous sections of this presentation. There are a number of open source libraries such as Interpret ML, AI Explainability 360, Alibi Explain, and Captum, which offer generic implementations of neuron and feature attribution algorithms. In this presentation, we will focus on Captum library and apply some of the algorithms from Captum library to different NLP problems. Captum is an open source library which offers techniques that help model developers to understand and debug their models. The library has three main characteristics. The first characteristic is the multimodality. This means that you can apply Captum to any type of PyTorch models and input stemming from different sources, whether it's image, text, audio, or video. Extensibility means that we can easily extend the library and add new approaches from state-of-the-art literature, and ease of use means that the library is designed to be used easily with a couple lines of code. The latest open source release of the library is 0.3.1, and it offers a number of gradient and perturbation-based attribution algorithms and evaluation metrics. The attribution algorithms can be grouped into three categories. The first category of attribution algorithms help us to attribute output predictions to the inputs of the model. The second group of the attribution algorithms allows us to attribute output predictions to all neurons in an internal layer. These are the approaches such as layer gradients, layer integrated gradients, or neuron ablation, as we also discussed in the previous slides. Lastly, the third group of attribution algorithms allows us to attribute any internal neuron to the inputs of the model. Apart from this, the library also offers two evaluation metrics in fidelity and sensitivity max. These two methods we also introduced in the previous section. On this slide, we summarize all attribution algorithms in Captum library. Some of these algorithms we have introduced in detail in previous slides, for example, integrated gradients or layer conductance. Note that layer attribution algorithms return attributions for all neurons in a given layer L. These are the algorithms that we will use to estimate neuron importance later in this presentation. The perturbation-based approaches are highlighted in pink, the gradient-based approaches are highlighted in purple, and the approaches that do not belong to any of these two groups are highlighted in dark blue. On my left side of the diagram, we see the list of algorithms that help us to attribute output predictions or internal neurons to the inputs of the model. On my right side, we see the list of the algorithms that help us to attribute output predictions to all neurons in an internal layer. So these are the approaches that we will use in the next slides. So now let's apply the attribution algorithms from Captum library to different real NLP problems. Let's assume that we're given a sentiment analysis model based on BERT network architecture that has 12 hidden layers and is trained on Stanford Sentiment Tree Bank dataset, also known as SST dataset. Let's also assume that we are making predictions using a pre-trained model and a given text sample, such as "it is such a wonderful movie." In addition to that, we also assume that our model is able to make correct prediction and correctly predict positive sentiment for this example. And our goal is to understand the role and the importance of different neurons in given layers for positive sentiment prediction. It is important to note that BERT architecture specifically allows us to have token level groupings of neurons in the output of embedding on all 12 BERT layers. This makes it easy to visualize a summary of all neurons for each token. This is possible because the outputs of embedding on all BERT layers have the output shape of number of examples by number of tokens by embedding dimension. So now let's compute the importance of neurons in the layer number 7 using gradients of output prediction with respect to the neurons in layer 7. We can use layer gradient time activation algorithm from Captum library and its attribute methods to attribute to all neurons in the layer 7 for predicted positive sentiment class, which is denoted by target index 1. The output of gradients have the same shape as the output of layer 7, and that shape is number of examples by number of tokens by the embedding dimension in the layer 7, which is 768. In order to be able to visualize neuron attributions for the tokens, we summarize the attributions for all neurons across the embedding dimension, which is we summarize the attribution scores for all 768 neurons for each token, and this gives us token level feature, token level importance. Now we can visualize the token level importance using the helper visualization functions from Captain Library. Tokens colored in red indicate the irrelevant tokens and for positive sentiment prediction, and tokens highlighted in green, they indicate the relevant tokens for prediction of a positive sentiment. The intensity of the color indicates the strength of an importance for the specific token for the positive prediction. So in this case, we did visualize for the layer 7, when we can see that we don't have anything highlighted in green, which means we don't have any tokens that are positively correlated with positive sentiment in this layer, but we can check out another layer and see how does importance look like for another layer. When we move to layer 10, then we can see that for layer 10, we are able to detect a positive signal highlighted in green for the class token. But class token is not that important for positive sentiment. Maybe still this layer learns that the class token could be useful for predicting positive sentiment. Now let's move to another algorithm, namely layer integrated gradients, and see what kind of importance signals the layers and neurons learn. Similar to previous algorithm, we call attribute on the instance of the algorithm, which in this case is layer integrated gradients, and we summarize the attribution for all 768 neurons per token so that we can visualize a summary of neuron attributions for each token. When we visualize the attributions for each token, we observe that the importance scores based on integrated gradients are more meaningful. The token wonderful appears to be most important according to layer 7. Now let's move to layer 10 and see how does the importance changes. When we move to layer 10, we can see that a token wonderful is still important, but not as important as class token. It appears that the neurons in the layer 10 are more focused on class token, as we also observed for saliency algorithm in our previous example. So in a general case, we can use any layer attribution algorithm from Captain Library to compute the importance of neurons in any given layer for a specific prediction. To do so, we need to create an instance of layer attribution algorithm and pass the forward function of our model, a list of layers where the neurons are located, and call attribute on that instance by passing our input examples and the target prediction index and other necessary arguments for a specific implementation. Now let's look into a case study for a question answering model trained on BERT architecture. For our case study, we fine-tuned BERT question answering model on squat dataset using Hugging Face library. This helped us to reach 86% F1 score and 78% exact match between prediction and ground truth. The goal of this case study is to understand the roles of different types of word tokens, layers for a model, and neurons for a model prediction. There exist a number of papers that aim to explain the inner workings of BERT models. Most of those papers analyze attention matrices in different heads and show different properties that those heads learn. For example, they show that some of the heads are focusing more on linguistic properties. There are also papers that argue the usefulness of attention matrices in terms of model explainability and focus on further transformations in vector norms. In this case study, we will focus on analyzing the neurons in different BERT layers and understand their importance for model prediction. Let's assume that we're given a text example such as, "It is important to us to include, empower, and support humans of all kinds." Let's also assume that we're given a question that is as simple as what is important to us. Our goal is to predict the start and end position of the answer to given question in the input text and analyze neuron importance for those predictions. Before we start making predictions, we need to concatenate question with the text and tokenize that. After tokenization, we perform prediction on tokenized text. This results to start position prediction as 2 token with a prediction score 0.52 and end position prediction as token kinds with a prediction score 0.75. Now let's explain the predictions of our model with the help of embedding on all 12 BERT layers. To do so, we place explanation hooks on the outputs of those layers and examine neuron importances for both start and end position prediction. In order to reach that objective programmatically, we need to load the pre-trained fine-tuned and pre-trained BERT model first, and that's what we do on this slide. Then we use layer integrated gradients to explain the output neurons of the BERT embedding layer. In order to use layer integrated gradients, we need to first define a forward function that returns either the start or end position prediction using position flag based on the original model's forward function. The attribution algorithm returns an attribution score for each neuron in the embedding layer. We summarize all 768 neurons for each token and visualize the attribution scores for each token for start position prediction. As we can see, there are multiple tokens that are important for the start position prediction, such as what, important, or is. The token as next to the start position prediction, token 2, has also a high importance score. When we attribute to the end position prediction token kinds, we can see that the question token what is still important, along with kinds token, which is predicted as the correct end position token. Similar to in BERT embedding layer, we can examine all 12 BERT layers using layer integrated gradients or layer conductance algorithm. In this case, we chose layer conductance algorithm. We set interpretation hooks on all 12 BERT layers in a loop. And in a loop, we compute attributions for all neurons in all 12 BERT layers, both for start and for end position prediction. In this plot, we zoom in into one of the tokens, namely token kinds, and examine the distribution of attribution scores across all 768 neurons for start position prediction. We can do the same for end position prediction and also for other tokens. We see here how the distribution of attribution scores changes across all 768 neurons in all layers. The median remains 0 for almost all layers, and the interquartile range reduces slowly as we move towards last layer. We can further analyze attribution scores by computing probability mass function for attributions in all 12 BERT layers. In this case, we distributed attribution scores between 100 bins as shown on the x-axis and visualize the density of attributions for all 12 layers on the y-axis. The distribution for most of the layers resembles bell curve with varying means and variance values. Now we can use probability mass function to compute Shannon entropy as an example. On the x-axis we visualize the layers and on the y-axis total attributions. The size of the blue circles for each layer total attribution pair corresponds to the normalization entropy value at that point. In this particular example, we observe that entropy doesn't change much from layer to layer. However, in a general case, entropy can provide an intuition about the distributional characteristics of attributions in each layer and can be useful, especially when comparing it across multiple tokens. Now, instead of looking into each token individually, we can summarize the neurons per token for all 12 bird layers and visualize them as a heat map. In this case, we visualize the heat maps for start position prediction. As we can see, the question token what has high importance for almost all layers, except the last 12th layer. Token it becomes more important in the last two layers, and correctly predicted token 2 also gains more importance when moving towards last layers. We perform the same experiment for end position token prediction. As we can see from the question, the question token what still remains very important for end position prediction. Also, token it gains more importance, especially in the last layers. And the correctly predicted end token kinds also appears to have high prediction score across all layers, which becomes more obvious, especially in the last layers. When we put start and end position prediction heat maps next to each other, we can see some commonalities. First of all, we observe that the question token what has similar importance patterns for both heat maps. We observe similar phenomenon also for the it token. For end position prediction, we can clearly see that some tokens in the end of the text become more important, such as humans and kinds. So this type of analysis helps us to understand how neuron importance changes from layer and layer, and potentially how it can be helpful to understand our models even better. In this part of the tutorial, we connect two different worlds that we introduced throughout this tutorial: concept-based neuron analysis and neuron importance based on attribution for a specific model prediction. As you might recall from the previous parts of this tutorial, we introduced techniques that help us to perform fine-grained neuron analysis and identify what different concepts, such as concept of proper noun, our model learns, independent of predicted class. Later, we introduced techniques that help us to estimate the importance of all neurons in an intermediate layer L by attributing an output prediction to the neurons in a given layer. We consider connecting these two worlds. That is, we combine important neurons for a specific prediction and detected concepts for those neurons. This will help us to explain model predictions in terms of concepts with high neuron importance scores. It turns out that there already exist algorithms that are based on this idea of connecting neuron concepts with neuron importance based on attributions. One of those approaches is called quantitative testing with concept activation vectors, or TCAV. In order to explain how TCAV works, let me first talk a little bit about how the concepts are represented. Here we have examples of two concepts: positive adjectives and neutral terms. Positive adjectives concept comprises words or text that convey positive sentiment, such as beautiful, brilliant. Neutral terms concept consists of disconnected neutral terms such as sense, interesting, and is distinct from positive adjectives concept. TCAF helps us to measure the sensitivity of model predictions to different high-level concepts, such as positive adjectives or any other concepts we choose to. In order to be able to explain TCAF, let's take an example model, and let's say that we are given a pre-trained sentiment classification model which outputs a probability sigmoid score indicating whether the text has positive or negative sentiment. Let's also say that we are given predictions using text that contains positive sentiment based on the movie rating dataset. We have here two examples of two movie rating descriptions, but you can imagine that there are more than two examples. Let's assume that a model is being able to predict positive sentiment correctly for most of our input examples. Also, let's assume that we have predefined sets of concepts such as positive adjectives, as we introduced in the previous slides, and that we want to know whether the positive adjectives in the text played any role in predicting positive sentiment for those specific text examples. TCAF helps us to test the importance of different model layers for model prediction. It consists of two steps. The first step is the generation of the concept activation vectors, or the CAVs, which is concept learning for the neurons in the given layer, and the second step involves directional sensitivity computations, which is the attribution for a specific output prediction with respect to the neuron. Let's go over each of these steps and understand how these two steps procedure works. First, let's understand that what CAF means and how we can generate them. Let's assume that we are given a multi-layer neural network that performs classification. In this example, we have a deep neural network that takes an input features and produces a prediction score. Let's say, let's select a layer in the model and call it L. Let's also assume that we are given an FL function which computes the activation in the layer L, and HLK function that takes those activations and produces a prediction score for our model. This is similar to the definitions that we had for, in our previous section. In order to compute concept activation vectors, let's first compute the activations for all concepts that we have in given layer L. In this case, let's compute activations both for positive adjectives and neutral terms concepts using the FL function. Now let's train a linear classifier that helps us to distinguish between those two classes of concepts, positive adjectives and neutral terms. Concept activation vector, also known as CAV, represents the vector that is orthogonal to the hyperplane that separates those two classes, positive adjectives and neutral terms. For positive adjectives, the CAV vector is orthogonal to the layer hyperplane and points towards the positive adjectives concepts. For neutral terms concepts, CAV vector is also orthogonal to the hyperplane but points to the opposite direction. Note that binary class problem in this case is just an example. In a general case, we can have more than two concepts and more than two classes. Now, if we look back to the concept analysis using neuron probing by a linear classifier and CAV, we realize that they are very similar. In both cases, we train linear classifier to estimate concept relevance based on the learned weights of the linear model. There are certainly also some differences. For example, neuron probing classifier considers neurons across multiple layers when training the classifier, and CAV considers the neurons of one given layer L. Nonetheless, these two techniques share similar ideas. Now, after computing the CAFs, let's move to the second step and compute directional sensitivities. Let's recall from the previous slides that our goal is to identify which concepts are important for predicting positive sentiment in the text. For those reasons, we would like to understand the sensitivity, the gradients of the output positive sentiment prediction with respect to the outputs of our layer L, that is the neurons in layer L. In other words, we would like to compute the attribution of a specific prediction with respect to the neurons in layer L. This is exactly what we do in step 2. We denote the attributions of output positive sentiment score with respect to the outputs of layer L, ATTR subscript L superscript positive sentiment class. As next, we combine the outputs of those two steps, CAFs and attributions, together with a dot product, which results to a CAF underscore ATTR vector that describes the sensitivity of each positive sentiment prediction to the positive adjectives concept in the layer L. In order to compile and create one quantitative score and aggregate CAF underscore ATTR scores across all test examples, we divide the number of text examples with positive CAF ATTR scores by the total number of input examples. In other words, TCAF represents the fraction of input examples for which the sensitivity direction or the attribution is aligned with the CAF direction. In a general case, a TCAF score for a given class, layer, concept, and inputs is equal to the fraction of examples that have CAF and directional sensitivity vectors aligned. Note that instead of using number of examples with positive CAF underscore ATTR score, we can also use the magnitudes of CAF underscore ATTR scalar. It is important to mention then that TCAF can potentially learn meaningless CAFs for meaningful concepts, depending on the biases in our concept dataset or the linear model that learns the CAFs. Also, it can happen that CAF generated by the random or neutral concept can be meaningful depending on the neutral concept dataset definition. In order to mitigate those issues, we can test statistical significance of our concepts using two-sided statistical significance test. We can do that, for example, by testing a specific concept, for example, positive adjectives, against multiple neutral concepts. If positive adjectives concept is truly meaningful, it will stand out with high TCAF score among all neutral concepts. Now let's discuss some of the limitations of TCAF. TCAF requires predefined concepts, as we saw in our previous slides. We had to have a predefined set of positive adjectives and neutral concepts. This could potentially require some human labor, but it would be great if we could generate them automatically. Another problem is that CAFs can be meaningless. As I mentioned in the previous slides, two-sided significant tests can help us with that, but for that we need to compute TCAFs for a larger number of experimental sets or concepts, and this can be computationally and memory intensive. Now let's look into the results of our case study for sentiment analysis model using IMDb dataset. The model used for this experiment is a CNN model, which is also linked on this slide. Our goal is to explain hand-picked examples of positive movie rating using TCAF. Let's assume that we have already loaded our pre-trained sentiment analysis model and we are able to make predictions using test examples of movie rating descriptions. Let's also assume that we are given two sets of concepts, positive adjectives and neutral terms, as also described in the previous slides. Our goal is to apply TCAF on our model using these two concepts and understand how important they are for positive sentiment prediction. Note that our model has two convolutional layers and we ran TCAF for both convolutional layers. On this slide, we visualize TCAF scores on the y-axis for both convolutional layers and both concepts, positive adjectives and neutral terms. As we can see, positive adjectives concepts have a relatively higher TCAF score compared to neutral concepts. As next, we repeat that same experiment and use 5 different concept datasets for neutral concept in order to determine whether positive adjectives is indeed significantly more important than neutral concepts. As we can see, TCAF score for positive adjectives is indeed significantly higher compared to the negative terms across all 5 experimental sets. We then move ahead and plot the distribution of TCAF scores across all our experiments for both positive adjectives and neutral terms for one of the convolutional layers, conv2, using box plots. As we can see, positive adjectives have a larger mean and higher TCAF scores. In addition to that, we also perform two-sided significance tests in order to estimate whether those two distributions are different or not. In this case, low p-value indicates that those two distributions are indeed different and disjoint. We repeat the same experiment for convolutional layer 1, conv1. Here as well, we observe that positive adjectives and neutral terms concepts have disjoint distributions with small p-values, and that the mean of the positive adjectives concept is much larger compared to the one of neutral terms. TCAF is one of the approaches in concept-based model interpretability research. There are a number of other publications in this area of research. For example, one of them is trying to automatically extract concepts from images. This can help with the limitations of having predefined sets of concepts, and this type of approach can also be adapted for NLP problems. And not only for images. Another recent research publication aims to identify a sufficient set of concepts that describe our prediction. It adapts a sharp algorithm to concepts in order to identify most important and influential concepts. So with this, we would like to wrap up this tutorial and say thank you. We hope that you enjoyed watching our session on fine-grained interpretation and causation analysis in deep NLP models.
Thank you, Narine. Now I would like to present a high-level summary of what we have learned in this tutorial. First, we looked at various methods for fine-grained analysis, which tells how various language or task-specific concepts are learned and represented within neurons in the network. However, it does not tell whether those neurons play a role in models' prediction or not. On the other hand, causation analysis highlights which neurons were used in making a particular prediction, but it doesn't provide a definition of those neurons with respect to concept. In the last part, we have seen that combining the two leads way towards concept-based interpretation of models prediction. There are several open research questions and challenges in each type of analysis. For example, how to identify the group of neurons working together to learn a concept or work towards a prediction is still an open question. Similarly, overall basis of concept-based interpretation is relying on human-defined concepts. Do we know if our model rely on human-defined concepts? Can we identify concepts based on how model has learned about the task and the language? We hope to see the answers to many of these questions in the near future, as they are important to improve our interpretation of deep models. I would like to leave you here. Thank you very much for your attention. Have a good day.